Hello, welcome to Genomics for Dermatology. My name is Hennies, and I will provide you another part of our pre-course material. I will tell you something about the regulation of gene expression, about the transcriptome. My presentation has two parts. The first part, I will give you some background information about how gene expression is regulated on the DNA level. And in the second part, I will speak a little bit about the applications in the context of human health and disease. I have also listed some textbooks here. So if you want to go into more detail, these textbooks have a lot of background information about these topics. So you've heard about gene mutations, and obviously mutations would reproduce on the transcript on the level of the primary transcript and then be translated into the protein and lead to some cellular response. So we have to think about how the transcription takes place and how the transcription is regulated, that is then involved in the cellular response to mutations on the DNA level. So for the transcription of genes, we mainly have three different RNA polymerases. For the synthesis of our transcripts, we have three different RNA polymerases: polymerase 1, polymerase 2, and polymerase 3. For protein coding genes, which are transcribed into mRNAs, mainly RNA polymerase 2 is used. So I will focus on the RNA synthesis and RNA polymerase 2 activity on the following slides. RNA polymerase 2 recognizes core promoters. So each protein coding gene has promoter elements in front of the transcription start site. There are a number of elements of recognition sites of these different core promoters, including BRE, the transcription factor 2B recognition element, of course, the TATA box, which is recognized by the TATA binding protein that is part of the transcription factor 2D complex. Then we have a number of other elements like the initiator element and the downstream promoter element. All these are recognized by subunits of the transcription factor 2D. It's important to note that none of these elements is either sufficient or is obligatory for transcription. And we will see that the flexibility of these different, the flexible use of these different elements is important for the flexibility or the control of gene expression in eukaryotic cells. So how does the transcription basically takes place? This would be the initiation of transcription at the RNase polymerase 2 promoter. We have the transcription start site here. We have the TATA box in front of the transcription start site. This is recognized by transcription factor 2D and the TATA binding protein. And as soon as this and other transcription factors have bound to the promoter, the RNase polymerase 2 would be recruited, which binds, which is activated by some other factors, and which then recognizes the start site of the transcription and starts with the transcription, the synthesis of the RNA strand as soon as nucleotides are available and it is phosphorylated. So this is how transcription starts, which involves other recognition sites and other elements. So understanding about the binding sites for these various transcription factors is important for the understanding of RNA synthesis and the regulation. And this is one major task of the ENCODE project, to identify and to map these different recognition sites. And I will come back to ENCODE and the ENCODE project in the second part of this presentation. So we have the initiation of the transcription, and we of course also need a defined end of the transcription, and this is how the termination is realized. The RNA polymerase 2 would move along the gene and would synthesize the new RNA strand. At some point, there is the polyadenylation signal, which identifies the end of the transcription unit. And as soon as this polyadenylation signal is reached, is translated, is transcribed into RNA, cleavage takes place, and the complete RNA strand is removed. An exonuclease would then degrade the remaining tail of the RNA synthesis, and as soon as the exonuclease meets the polymerase, the transcription stops, and we have the RNA strand here separated from the polymerase. So this is the basic process of RNA transcription. We need the promoter elements to recognize the transcription start site, but transcription needs a number of other elements for the activation of transcription. So we have the core promoter here, which binds the polymerase II, and we have the Teta box, the initiator element, and the downstream elements, which are all part of this control unit. But there are other elements that are also involved. This has been identified in recent years. Actually, there are clusters of other recognition sites, which are called locus control regions. And these involves enhancers or silencers. So we could have a positive or a negative control. And these enhancer elements could be several kilobases or even megabases apart from the target gene. They are identified again by transcription factors, which then interact with cofactors, and all these together would lead to the activation of transcription of the target gene. So we have a core promoter with a number of promoter elements of recognition sites, and this would be the so-called canonical core promoter with some of its elements. This is in a region which is open on the chromatin level, so it is free of nucleosomes. We have nucleosomes on either side of this core promoter. However, this would not really explain the flexible situation that we have identified in recent years, because transcription takes place not just from one particular site, but most genes have multiple transcription start sites, which makes transcription more flexible depending on developmental status, depending on cell type. So we would, instead of the canonical promoter elements for most eukaryotic genes, we would rather have a transcription initiation platform, which is flexible for multiple transcription start sites. How would this be realized on the molecular level? We need some interaction between the remote control, between the enhancer elements and the core promoter with the polymerase II. The initial idea was that there's a physical proximity where the DNA forms such a loop, and then the enhancer element and the transcription factors bound to the enhancer elements would interact with the polymerase II and the transcription start complex. This static protein-protein interaction would, however, not explain how the flexibility is achieved and how, for instance, multiple transcription start sites can be activated depending on a particular cellular situation. And so the recent model, the current model, is rather a flexible model where the enhancer elements, the cis-regulatory enhancer elements, bind transcription factors and cofactors. So there's an interaction between cis elements and trans-regulatory factors, and they form such a transcription activating microenvironment. So we have an enhancer here. This enhancer would interact with several transcription factors and other elements, which then lead to the activation of the promoter and the polymerase II. This would be a dynamic protein-protein interaction, which can respond to different cellular situations. So this would explain the activation of multiple transcription start sites. However, it does not yet explain how the enhancer recognizes the target gene. How does the enhancer know which target gene would be activated? So we need a higher level organization of the chromatin in order to realize this, this control. And these are the so-called topologically associated domains. So each of these pyramids here represents one topological associated domain, one functional unit of the chromatin. And the enhancer in one of these units would then just activate the gene which is in the same unit. And this means insulators are needed that separate each of these units. And these are recognized by insulator proteins such as CTCF. Mapping of these CTCF sites on the chromatin, on the DNA level, would thus identify the boundaries of the domains and would identify the genes which are controlled by particular enhancers. There's one further element which is important for the control of gene expression, and this is the DNA accessibility. Genes are not just activated depending on the assignment to particular domains, but they can also be activated or not activated depending on accessibility of the DNA. So the enhancer would only activate a gene where the DNA is accessible. And this accessibility is achieved by different structures of the chromatin, either an open chromatin structures or a condensed chromatin structure. And this structure is mainly determined by the nucleosomes. So we have to have a look at these nucleosomes. This is one such nucleosome, which consists of 2 turns of DNA around the core protein unit. And this core unit consists of an octamer of 4 histones to each of H2, H3A, H3B, and H4. Each of these histones has a positively charged N-terminal tail, and these tails can be further modified. And this modification is again important for the control of gene expression and the activity. Some of these modifications are, and most of these modifications are on lysine residues, and they can be either acetylation or methylation. In this example, this would be a trimethylation on a lysine residue. So we would have a map of these modifications. This would be the sequence of the histone tail, and we have various modifications, methylations and acetylations, and some other modifications that are characteristic for this particular situation. So to summarize this control of the gene expression, we have a variety of different modifications and different levels where the expression, the activation of the promoter is controlled. We have in the inactive state, we have a frequent methylation on the DNA level. We have a characteristic pattern, a signature of modifications on the histone tails. And we have the condensed, the closed structure of the chromatin. In contrast, in the active situation, there's only few methylation. There are only few methylation sites on the DNA level. There's a different pattern of modifications on the histones, and we have the open chromatin structure where the DNA is accessible for transcription factors. And this gives the flexibility that is recognized by is needed for the cellular response to particular situations in the control of gene expression and transcription.